Thank you, the conference, for inviting me, the organizers. They're great, and every year I come here and have a lot of fun. My name is Marek Kubicki, and I am professor of psychiatry and radiology at Harvard Medical School. But I'm also trained radiologist and MR physicist. So over the last 20-25 years in my research, I was interested in neuroimaging. I was using neuroimaging to understand mental disorders. Most of my studies are focusing on schizophrenia, but we're also investigating bipolar disorder, other mood disorders, depression. Recently, we started working with the cohort of eating disorder patients. And at the very beginning of my research, I was just using neuroimaging to find differences between groups and to try to understand better where they're located and why there and nowhere else, or what is the difference between the location of the lesions between different diseases. But gradually over time, we started being interested more in the biology of those disorders, or in the biological explanation of the changes that we see on neuroimaging. And about 10 years ago, we started being fascinated by the concept of neuroinflammation in the brain and as part of those disorders. And we started developing methods, and we started looking closer at the mental disorders, and especially focusing on neuroinflammation. So today, I'm going to be talking in general more about neuroinflammation than about schizophrenia, and I will be talking about neuroimaging, but I will also try to get a little larger picture on neuroinflammation, other biomarkers that we have available, and maybe also mechanisms and how potentially we could treat these biological phenomena in the mental disorders. So first of all, what is neuroinflammation, right? And if you talk and ask immunologists, they would have different explanation and different definition of neuroinflammation from neuroscientists, from biological scientists, and neuroimager. But to me, neuroinflammation is really the disease of a central nervous system that involves activation of the glial cells. So if the injury or other factor activates microglia and astrocytes, activates the cells, the glial cells in the brain nervous system, then we have the neuroinflammatory process. Neuroinflammation has been investigated for a long time, and maybe around 20 years ago, it was rediscovered and started reintroducing to the realm of mental disorders, and that is more or less the time when the work on biomarkers began, and I'm going to show you more about where we are right now. So again, the key players of neuroinflammation are, I'm going to try to show it. This is the microglia. They are the primary immune cells in the brain that monitor and respond to pathological conditions. The astrocytes that support neurons and regulate the inflammatory processes. They are also involved in permeability of the brain-blood barrier. And the cytokines, which are signaling molecules that mediate the inflammatory responses and can affect neuronal function and health. There are several different mechanisms of neuroinflammation and potential causes of neuroinflammation. The potential causes include infections, viral, bacterial, trauma, toxins, and environmental toxins, as well as substance abuse and stress. especially in the traumatic brain injury. And in response to pathogens, injury, or stress, glial cells become activated, leading to release of inflammatory mediators. Now, neuroinflammation, as I mentioned, has been implicated in the pathophysiology of various mental disorders, and inflammatory response in the brain may disrupt neurotransmitter functioning and synaptic plasticity, and also affect neurodevelopment, brain development, especially in white matter, and that could lead to symptoms of mental disorders. And the disorders, this is not the entire list, but these are disorders that where the inflammation has been implicated to a larger or smaller degree. Those include depression, schizophrenia, bipolar disorder, PTSD, autism spectrum disorders, and eating disorders. So going through the list in terms of depression, the role of cytokines. The cytokines are involved in depression, and their increased levels of inflammatory markers can lead to alterations in neurotransmitter systems, such as serotonin and dopamine, and those contribute to the development of depressive disorders. The cytokine that has been mostly implicated in bipolar disorder is brain-derived neurotrophic factor, or BDNF. And BDNF is involved in development and neuroplasticity. And the inhibition of the BDNF, of the production of BDNF, has been shown to aggravate depressive symptoms. In schizophrenia, neuroinflammation has been shown to be involved in brain development. There's a prominent theory called two-hit theory, where the first hit involves the early neurodevelopmental abnormality that makes the brain vulnerable to the second hit. And recently it's been believed that the second hit could also be oxidative stress and following neuroinflammation. And obviously the chronic neuroinflammation can cause additional damage to the brain and can cause progressive worsening of the psychotic symptoms and deterioration of the cognition. In bipolar disorder, inflammation may play a role in cycling of mood states seen in bipolar disorder, and cytokines and BDNF have been similarly to depression linked to neuroplasticity and cognitive deficits. In PTSD, the mechanism is a little different, or seems to be a little different. The traumatic events can exhibit an inflammatory response, primary in areas related to emotion regulation, and chronic inflammation might influence the brain's stress response system, potentially leading to heightened fear and anxiety in patients with PTSD. Also, in eating disorders, neuroinflammation has been shown to be involved, and here the neuroinflammation can cause hormonal imbalance, mood regulation, as well as changes in appetite. So far we've gotten relatively good at measuring inflammatory markers outside of the brain, in the periphery. In the blood, we draw the blood, and we run either the panel that is focused on several cytokines or interleukins, or we can do something that I highlight here. It's called proteomics. It's just a big panel of over 5,000 proteins that can be measured from one sample, and the reason why we're focusing on proteins in the blood is because they are executors of most biological processes. They are dynamic indicators of phenotype in health and disease, and they provide the most actionable target for therapeutic interventions. But when we're measuring 5,000 proteins, we obviously have a lot of proteins that might be involved in a single biological process. In fact, these are the processes and the number of proteins that are involved in each of those processes, and if each of those processes that we list here are somehow related to inflammation. So you can see that the outcome of the proteomics analysis in this case is in hundreds of proteins that might or might not be involved. Then once we have that many proteins, it is important to understand the pathways that those proteins are involved in. It's not just simple analysis of a single cytokine. You have to kind of understand what the certain proteins are, what process they're involved in, and what cell type or immunocell type they are activated by, and then you can understand better the pathology. But okay, we know that we can measure them in the peripheral blood, but how does it relate to neuroinflammation, to whatever is happening in the brain? So based on several theories as well as preclinical research, we know that under healthy circumstances, blood-brain barrier is quite impenetrable to pathogens. However, when there's an inflammation in the periphery, depending on how many pathogens are involved in this inflammation, the blood-brain barrier becomes leaky. And there are certain disorders where the blood-brain barrier is the primary source of inflammation going through from the blood to the brain. And in MS, for example, the blood-brain barrier is so leaky that the contrast agent that we inject during MRI goes through the blood-brain barrier and enhances the lesion very well, so that we can see it with the naked eye. In mental disorders, it's probably not the case. It's most likely that the blood-brain barrier is a little leakier than in healthy condition, and with time, either the cytokines themselves, or if the problem becomes bigger, the larger immune cells can migrate through the blood-brain barrier, activate the glial cells, and it sort of causes the vicious cycle where glial cells produce their own cytokines, which go back to the blood-brain barrier, can be detected with proteomics, but also exaggerate the inflammatory cascade. As far as neuroinflammation in mental disorders, obviously the evidence is mixed. There are some studies that are showing such a big differences, like this one from 2009, published in Molecular Psychiatry. The authors measured interleukin-1 beta in the periphery, but also in cerebrospinal fluid, and this is really striking, the difference between patients with schizophrenia and healthy controls. This is another example of this big difference. This is the component C4, also involved in activating microglia and neurodevelopment. And those studies really renewed our interest in neuroinflammation. There are some other studies that when we started being involved, we started collaborating with other people from other diseases, not just focusing on schizophrenia. And this is the slide that I got from my collaborator from New York. Catherine Millet, who is focusing on a bipolar disorder, and she's looking at peripheral CRP, C-reactive protein, and she published a paper showing then that there is a difference between CRP during mania and depression comparing to control and condition. And she also showed that the CSF and plasma CRP are very correlated, indicating that what I just said, once the peripheral neuroinflammation goes into the brain, that the vicious cycle sort of equalizes the measurements, and what we have in the brain can be measured in the periphery. She also showed that CRP levels are related to cognitive decline, so more CRP, the larger cognitive decline. My collaborator working on anorexia nervosa in eating disorders, Lauren Breithaupt, published this study. This is the proteomic study that shows a lot of inflammatory related proteins that are upregulated in anorexia comparing to healthy controls. So there is evidence, obviously, that mental disorders, or at least subset of people with mental disorders, present themselves with increased levels of inflammatory markers in the periphery. But since we're doing neuroimaging, we started working on biomarkers, and we're wondering what is the best way to visualize neuroinflammation in the brain. And thus far, the field is still developing, and the methods are coming up, and they're tested, and some of them are not being replicated, or findings are not being replicated. But in general, there are three ways of measuring neuroinflammation in the brain. One is directly trying to bind your PET ligand to the either activated microglia or activated astrocyte. Those methods, the majority of them are just binding to or majority of the ligands are binding to microglia. Some of them are binding to both. TSPO is the prime example of such a method. Another way of looking at neuroinflammation is to look at the permeability of the blood-brain barrier, because, like I said, the theory is that if there's peripheral inflammation, the blood-brain barrier becomes more leaky, and either contrast goes through it or the water goes through it, and one can measure this perfusion. And the third way of looking at neuroinflammation is obviously looking at which is more or less what we have been doing all the time. We're looking at the potential consequences of neuroinflammation, whether it's a damage to the myelin or damage to the axons and brain degeneration. So let me go through some of those methodologies and show how they work and what has been found so far. This is the dynamic contrast enhancement, and the way it works is it's basically the same way as the diagnostic MRI with contrast injection would work, except that since the leakage is much more subtle, you have to use more sensitive methodologies to measure the leakage or to measure the enhancement, and that's why you're scanning very rapidly in the first 15-20 seconds after the injection, and comparing the images. And this is the example of the study done in bipolar disorder, I believe. And this is the output of the methodology. So the red areas are showing where the contrast enhanced the image, and like you can see that there's at least 6 people that showed this enhancement. Overall, in the entire studied population of 36 people, 10 showed enhancement, 26 didn't. And that's consistent with other diseases. In schizophrenia, the similar studies showed about 30–35% people with enhancement comparing to 65 where their brains did not change from before to after the contrast. Another method that I mentioned is called TSPO. This is the PET method, and the ligand is binding to the mitochondria. When it's activating, there's more binding, and obviously the inflammation is a very energetic process that activates the ATP, and TSPO gets more binded to the activated microglia and astrocytes. And this is one of the studies that showed really very striking results and differences between healthy controls, and this is PET expression, this is TSPO expression in healthy controls, people at high risk to develop psychosis, and finally the schizophrenia patients. Another method that we're using in our laboratory is called free water, and I presented it a couple of times here already. It's basically tagging the water, extracellular water, and the idea is that in the inflammation, there's more water coming into the brain, there's edema in the brain, and we can measure it through free water. And indeed, we performed several studies where we found increased free water in patients with schizophrenia, both first episode and chronics, comparing to healthy controls. This is the blue areas are all the areas with increased free water. So we know that there are several potential methodologies that could be used to measure neuroinflammation. However, none of these methods is 100% proof. There are studies that report great results, but there are also studies that report no results, no differences between groups. I think the field is still at the stage where we're working on those biomarkers rather than have biomarkers to tag the subjects and assign them to the treatment. However, the hope is that once we have the biomarkers where we could stratify people into those that should receive additional anti-inflammatory treatment versus those that do not qualify for this treatment. There are some studies suggesting that non-steroidal anti-inflammatory drugs might work, or cytokine inhibitors. There are also some lifestyle intervention that have shown to be somewhat effective in decreasing inflammation. Those include diet, omega-3 acid, antioxidants, exercise, mindfulness, and stress reduction, to name just a few. So this is where the field is right now. I believe that we're pretty sure that there is a relationship between peripheral neuroinflammation and at least cognition, if not clinical symptoms. We know that neuroinflammation is or is very likely present in several mental disorders. We suspect that the mechanisms of this process might differ from disease to disease, and this is where it's important to understand the pathways and to study more the proteomics in this regard. We know that the peripheral biomarkers are better established, while the central biomarkers still require more research and validation. We are still not sure whether the neuroinflammation is a cause of clinical symptoms or whether it's a consequence of the disease. This is why we need to develop better the biomarkers And then we need to do some more clinical trials with drugs that modulate the immune response. So this is it. I want to thank all my collaborators. This is the work that is ongoing and includes multiple places, including Switzerland and New York. It wouldn't be possible without our sponsors and funding, and all the people that I listed on the Boston team. And that's it. Thank you.